So in this session we'll see about the structure of the antibody. So antibody, it is a Y-shaped structure. It is having a Y-shaped structure. As you can see, this is a Y-shaped structure. And of course, I have already told you it is a glycoprotein. So protein means it will be having an N-terminal end or the NH2 end. This is the N-terminal end and the carboxy or C-terminal end. And this Y-shaped structure, it is having two heavy chains. Two heavy chains. So here you can see this is the heavy chain and this is the heavy chain number 2. So they have two heavy chains. Heavy chain. And of course it has two light chain. Two light chains. And based on the heavy chains, this antibody, they are classified under 5 types. So they are IgG, immunoglobulin G, and that is having the heavy chain gamma. Then another one IgM, that is having the heavy chain mu. Another one IgD, that is having the heavy chain delta. Then you have the heavy immunoglobulin E, that is having the heavy chain epsilon, and the immunoglobulin A, that is having the heavy chain alpha. So these are the different types of immunoglobulin based on the heavy chain. And of course you have two light chain also. So these are the light chains. So these are the light chains. These are the light chains. And based on the light chains also, they are divided into two types, that is the kappa and the lambda. So this heavy chain naming, it actually means the heavy chain, the structure, that is heavy chain, it is actually due to the molecular weight of this one. So heavy chains will be having a molecular weight of around 50,000 Dalton, whereas light chain will be exactly half the molecular weight of heavy chain, and it will be 25,000 Dalton. So that is how the heavy chain and light chain. Then of course we have told that it is having an N-terminal and the C-terminal end. And of course we can see a carbohydrate thing. It is a glycoprotein. So you can see a carbohydrate attached to this one. This is the carbohydrate. You can just look like this. So this is the carbohydrate attached to the immunoglobulin. This carbohydrate helps to distinguish between two immunoglobulins or two types. So this is the carbohydrate. Helps in distinguishing between one immunoglobulin from another. Distinguishing one from another. Okay, so then we'll see another structural importance, that is this one. This region is known as the fragment antigen binding. So this is the antigen binding region. So this one is the antigen binding region of heavy chain. So that is FAB, fragment antigen binding. Here also you can see fragment antigen binding, and this one is the fragment antigen binding of the light chain. And here you can see this one is the FC region or fragment crystallizable. Fragment crystallizable and the fragment antigen binding. And this whole antibody, the entire structure won't go and attach to the epitope of the antigen. Only a certain portion goes and attaches to the antigen. And this portion here also, this portion, this is the variable region. This is the variable region of the light chain, and this is the variable region of the heavy chain. So this is the region. So this one is the variable region, and this one is the variable region. So this is the variable H, and this is the variable L. Variable light chain and variable H chain. And this region and the remaining region, they are the constant region. So here we have the constant H1. Here you have the constant H1, and this region, this is the constant H2, and this is the constant H3. So the heavy chain is having three constant region in a single arm, and here also the same thing. And of course the light chain is also having the constant region, that is known as the constant L. So an antibody structure, they are having the variable region and the constant region. This variable region changes. So that is how the specificity of the antibody antigen reaction happens. So this is specific to a particular antigen. There are variations in the antibody based on the antigen to which they are fitting. Okay, that's the thing. So another important thing I just want to tell you is that many interactions stabilize this antibody structure. They are having non-covalent interactions, as well as covalent interactions. And the non-covalent interactions includes H bonds or hydrogen bonds, then hydrophobic interaction, then of course van der Waals forces, etc. So these are the non-covalent interaction, and they also have a covalent interaction named disulfide bridges. That is very important because this is the one thing that stabilizes this entire antibody structure. So the disulfide interactions, you can see the disulfide interactions here. Here you can see two disulfide interactions. Here also you can see interaction. So these are the two disulfide interaction between two heavy chains, and between heavy and light chain also you can see the disulfide interaction. So here also you can see the disulfide interaction. So these disulfide interactions stabilizes the entire antibody structure. Okay. Then one more important thing you just want to notice is that there is a region known as hinge region. Hinge region, this is the hinge region. It's just the hinge region of the door. This is less like the hinge region of our doors. It is how the door is mainly fixed and how it moves over the hinge region. Likewise, a little bit flexibility is provided by this hinge region. This is the hinge region. So that is about the structure of the antibody with a Y-shaped structure, a glycoprotein with a Y-shaped structure which goes and binds to the antigen, epitope of the antigen. Okay, thank you.